I think I'll start by introducing you, Carolyn. So it's really nice to have Carolyn Guo here today to talk to us from UCLA. Carolyn did her got her MD at USC at the Keck School, and then has been at UCLA pretty much since that time. Did her pediatric internship and residency there, and then was a fellow in allergy immunology in the UCLA program, and then has been working extensively in the lab there since her fellowship, and she's done a lot of work on site-specific gene editing in hematopoietic cells, which she's going to talk to us about today. But she's also been involved in lots of other things that are of interest to us. I know she's been a PI on a couple of the lentiviral gene therapy trials, including, I think, most recently the LAD trial, and then also on the X-linked CGD trial, I think, as well, right, Carolyn?
Yes.
And has lots of expertise in immune deficiency diseases, and also has done some work recently on BTK gene editing as well. And I'm not sure what all she's going to tell us about today, but looking forward to hearing your talk, Carolyn.
Thank you very much for that introduction and for inviting me to speak today. Oops. Okay, so I have no disclosures, but I always like to start off with a picture of the hematopoietic tree. And as you may know, hematopoietic stem cell transplant has been utilized for primary immune deficiency since the 1950s. And it's improved significantly since then. But the reason that this is, has been curative for many PID is because of this multipotent hematopoietic stem cell, which is self-renewing and can give rise to all downstream lineages. And so if you replace hematopoietic stem cells of an affected individual with stem cells from a healthy donor, you could replace essentially their immune system and any cell that's defective downstream. However, the major limitations of allogeneic stem cell transplant has been the unwanted immune response between a patient and transplanted cells. And so a patient can either reject the transplanted hematopoietic stem cells, or donor T cells within the marrow can react against the recipient in graft versus host disease. And so when we are taking care of patients who have undergone allogeneic transplant, we're often dealing with this, trying to balance this yin and yang of GVHD and rejection. And so thus was born the idea of, what if we can avoid some of these challenges by using autologous transplant, so using a patient's own stem cells that have been corrected through gene therapy. And the way that this works is that if you know the gene that is responsible for an individual's disease, and you know the normal sequence for that gene, you can introduce it to their own hematopoietic stem cells, which could then engraft and give rise to all downstream lineages. So the main hypothesis driving this work has been that gene therapy using autologous hematopoietic stem cells that are corrected with the normal gene will have beneficial effects on blood cell production or function without the immunologic complications of allogeneic stem cell transplant. And the main benefits of this are that everyone would have a donor because it would be themselves. Autologous cells should not be immunogenic, and which also means that there should be no rejection or graft versus host disease. And immune suppression oftentimes would not be needed with conditioning or GVHD prophylaxis and treatment. And so practically what this would look like is you would harvest hematopoietic stem cells from an individual, either a baby or an adult. These stem cells are then taken to the lab where they are purified for the most, or for pure stem cells, which we use CD34 as a surface marker. And then the gene is fixed or added. Cells are formulated and then reinfused into the patient. It used to be that this process had to be perfectly timed so that while cells are in the lab, patients would receive cytoreduction with enough time for a washout period before those cells that had been gene modified would be freshly infused. And so this was, this was done for many patients and was successful, but it was also uncertain what we were infusing back into patients because it was given as a fresh product. And with advancements in cryopreservation, we've essentially been able to put a pause button in this process. And so now patients oftentimes are brought in for collection of cells, and they're sent home for months while cells are modified in the lab and then frozen, and then and while that product is being certified. And so it's not until the product is fully certified that patients are brought back for cytoreduction and reinfusion of the cells. We've also learned from many of our early gene therapy trials that that patients do need some degree of myeloablative conditioning. And the basic foundation for this is that if a patient's bone marrow stem cells are full of these red and gold stem cells, and you don't give them conditioning, there's not much room for new cells to engraft, even if they're their own. And so you can, take a mix of either full myeloablation or reduced intensity conditioning, depending on the disease process, and apply that when you treat a patient using gene therapy so that you can create room in their marrow niche. And so traditionally, gene therapy has been achieved through viral delivery of therapeutic DNA. And so these viral vectors have been stripped of their virulence factors, but still possess the ability to carry a DNA cargo and the ability to enter cells. And so upon entry to the cell, they can undergo reverse transcription, entry into the nucleus, and integration into chromosomal DNA. And the benefits of that is that this would result in expression of therapeutic mRNA and protein, but also that this integrated DNA would be stably transmitted to all progeny cells. So the main pitfalls of this form of traditional gene therapy is that expression of the gene is not normally regulated. And so when a viral vector is used to deliver a DNA cassette, that therapeutic gene is integrated randomly into the genome. And so it lacks the endogenous regulatory elements and promoter elements that tell it when to turn on and when to turn off. And so this approach typically relies on either an internal promoter that you include in the transgene, or the long terminal repeats of the virus to drive its expression. And many times the therapeutic gene is constitutively on. In addition, the viral vector can integrate near a proto-oncogene and turn on its expression, leading to cancer. And the way that this happens is through what we call polyclonal transduction, but eventual monoclonal expansion. And so if you imagine that this is your pool of stem cells that you're trying to treat, when you transduce it with a vector, it does not get into every cell, but let's say it gets into 3 of these cells. And there's a polyclonal transduction in that the vector enters the genome in different areas of the different chromosomes. So certain cells may receive one copy of a vector in one location, another cell in a different location, and some cells may receive more than one copy of the vector. And for simplicity's sake, let's assume that the cells that do not receive a vector won't produce a productive immune system. So let's pretend they die, so they don't contribute to the immune system. But the cells that do receive the vector can engraft and undergo lymphopoiesis and eventually reconstitute the immune system. However, if one of these clones happens to integrate near a proto-oncogene or a growth survival gene, there may be a monoclonal expansion of that particular clone, and which may be okay until some later point in which there is leukemic transformation and downstream disease. And this, unfortunately, is what happened in many of the early clinical trials in gene therapy for primary immune deficiency. So fortunately, it has not happened very frequently for ADA SCID, but you can see that for Wiskeld-Aldrich, chronic granulomatous disease, and X-linked SCID, that many of the patients treated with retroviral vectors when this first came out developed leukemia. The nice thing is that a lot of these issues have been resolved with the newer generation of lentiviral vectors. And so whereas retroviral vectors have a predilection for integrating near transcription start sites, lentiviral vectors tend not to behave in that way. And so because of that, despite these early poor outcomes with retroviral vectors, you can see that there have been many clinical trials that have come to fruition since then. And so although the rest of my talk will be focusing on gene editing for hematopoietic stem cells, I will say that there still is a place for viral vector mediated gene therapy. And for many children and adults, this has been life-saving. And so this is an example that I like to show of a 6-month-old boy who came to us at UCLA with ADA SCID. And so he is 6 months old, but he is at 50th percentile for a 1-month-old. So, we rarely see SCID babies present like this anymore. One, because we have newborn screening, but also because we have early intervention. And so now many of our babies who have severe combined immune deficiency who undergo gene therapy look like this. You can never distinguish them from any other healthy child. But so to overcome some of these pitfalls of viral vector mediated gene therapy came about an era of programmable nucleases. So this includes zinc finger nucleases, TALENs, CRISPR-Cas9, which I'll go into more. But zinc finger nucleases and TALENs are similar in that their DNA recognition is based on a DNA protein interaction. So in the case of ZFNs, their DNA specificity is defined by these arrays of synthetic zinc fingers that have been designed to interact with 3 to 4 base pairs of DNA at a time. And so you can imagine that if you string together a series of these zinc fingers, they can bind to a specific sequence of DNA. And so zinc finger nucleases and TALENs both work with 2 arms, both fuse to the catalytic domain of a FOC1 nuclease, which upon dimerization can create a double-stranded break. TALENs are similar, but instead of using zinc fingers, they use an array of amino acid repeats called repeat variable diresidues. They differ at these positions 12 and 13, which are specific for of a DNA base pair. And so similar to zinc fingers, you can string together a series of these RBDs and have it target a gene of interest. Subsequent to that and now we're in the era of CRISPR-Cas9. And so how this is different from zinc finger nucleases and TALENs is that their DNA recognition is based on an RNA molecule that directly hybridizes with DNA. And so if you imagine that this is your DNA target with all the ends shown in a row, you could create a short guide RNA that is complementary to your target sequence. This guide RNA is fused to a chimeric scaffold, which along with a protospacer adjacent motif, in this case, I'm showing the NGG PAM, will recruit Cas9 nuclease. And so this nuclease will create a double-stranded break exactly 3 base pairs upstream of the PAM. And so I just very quickly show you the difference between the CRISPR-Cas9 platform and ZFNs and TALENs. And so whereas ZFNs and TALENs are these relatively large helicoproteins that have to wind around the major groove of DNA to recognize its DNA base pair, CRISPR-Cas9 is essentially consisted of a short guide RNA that will zip through DNA and to look for its target. And so you can imagine that this, could be a more efficient system and could overcome some of the steric hindrances that you may encounter with ZFNs and TALENs, on chromatin structure. Sorry. So to understand how we can utilize gene editing, or utilize these technologies for gene editing, we have to understand how a DNA double-stranded break is repaired. So the most common route is through non-homologous end joining. So in general, when we have a DNA double-stranded break, our cell will just very quickly try to fuse that break back together to avoid additional loss of DNA base pairs. And so I consider that sort of a Band-Aid approach. It won't fix the gene, but it will prevent further loss. However, in certain phases of the cell cycle, particularly in S and G2, when there is the presence of a sister chromatid, when there is a DNA double-stranded break, it can instead use the sister chromatid as a DNA repair template and undergo homologous recombination. And so this will result in a base perfect repair of the broken DNA strand. And this is what we are harnessing when we try to perform gene editing on our primary cells. And so overall, what this would look like is you can cut DNA with ZFNs, TALENs, or CRISPR-Cas9. This results in a double-stranded break that can be repaired through non-homologous end joining, which essentially will break the gene and can be used for other purposes. Or if you provide the cell with a donor DNA template, so in this case, it's the blue part. So if that's your therapeutic gene cassette, and you flank that donor cassette with homology arms that parallel the area surrounding the cut site, so shown in the purple, you could essentially trick the cell into thinking that that is a sister chromatid and have it undergo homologous recombination. And so gene editing has been attempted and is being studied for many primary immune deficiencies. And I haven't listed all of them here, but these are some of the ones that have come about recently. But I'm going to focus a little bit more on our work for X-linked hyper-IgM, which is a defect in CD40 ligand. And so the X-linked hyper-IgM syndrome is a genetic defect characterized by the inability to switch from production of antibodies of the IgM to antibodies of IgG, IgA, or IgE types. And so in order for a B cell to produce class-switched immunoglobulin, it has to interact with a T cell. And an absolute requirement is interaction between CD40, which is constitutively expressed on B cells, and CD40 ligand, which is expressed by T cells only upon immune activation. And so defects in CD40 ligand are what account for the X-linked form of disease. And because CD40 is expressed on other cell types, such as dendritic cells and macrophages, patients present not just with an antibody deficiency, but with a combined immune deficiency. So they can have recurrent bacterial sinopulmonary infections, invasive pneumonias with 40% presenting with pneumocystis pneumonias. They can have complicated gastrointestinal infections, oftentimes with cryptosporidial species. They also suffer from autoimmunity and cancers. So on the left is showing a survival probability curve from a study of probably the largest collection of X-linked hyper-IgM patients published by Maite Della Morena. And as you can see, even with modern medicine, that the survival probability for these patients remains relatively poor, with about 30% survival at 30 years of age. We know that allogeneic bone marrow transplant is curative. However, there are several concerns, including unstandardized transplant timing and conditioning regimens, because this is such a rare disease. By the time patients go to transplant, many of them have had chronic infections, and so we worry about reactivation of these infections. There can also be worsening of pre-existing lung damage from prior infections, as well as these patients tend to have many issues with graft versus host disease. And so this is a schematic of the CD40 ligand gene. And as you can see, there are 5 exons, and all known disease-causing mutations are located within a single gene. And so this makes this an ideal candidate for gene therapy. And in fact, there were 2 groups that previously looked at the role of viral vector-mediated gene therapy for X-linked hyper-IgM. And both groups discovered that even though they could successfully deliver normal murine CD40 ligand cDNA using retroviral vectors, that 2/3 of mice in that top study and 1/3 of mice in the bottom study develop abnormal lymphoproliferation. And so this really emphasized to us that CD40 ligand expression needs to be regulated and cannot be on all the time. And this makes this particular disease amenable to gene editing. And so our rationale was that CD40 ligand gene is tightly regulated and requires expression in its normal chromosomal context. And we hypothesized that site-specific gene modification of the CD40 ligand gene in human hematopoietic stem cells will correct XHIM by autologous transplantation. And this can be done using site-specific endonucleases that target specific DNA sequences for gene modification and ultimately allow physiologic expression of the corrected endogenous CD40 ligand gene. And so just a quick schematic of our approach is that we would design nucleases that target the 5' UTR of the CD40 ligand gene. This would result in a double-stranded break that can be repaired either through non-homologous end joining, which is not our goal, or if you utilize the sequence surrounding the cut site as homology arms to flank a copy of the normal human CD40 ligand cDNA. If you provide that donor cassette to the cell at the same time, you could potentially allow homology-directed repair, integration of that corrective cassette at its endogenous locus under endogenous promoter elements. And so a lot of our early work was to compare TALENs and the CRISPR-Cas9 platform to achieve site-specific gene editing. And for this initial work, we designed TALENs as well as CRISPR guides that bound within the 5' untranslated region of the gene. And sort of, I'll just sort of jump to the conclusion of this work is, the take-home message is we felt that CRISPR-Cas9 worked better. So from the perspective of percent gene integration in primary human peripheral blood stem cells on the left, you can see that CRISPR-treated cells had higher rates of integration compared to TALEN. And then when we looked at the specificity of both platforms, even though we went into this thinking that TALENs may have higher specificity because it requires 2 arms, we actually found that there was no off-target activity with the guide that we were using, while a GFP IDLV trapping assay looking for off-target activity for TALENs identified 3 off-target sites, which we could detect in various cell types below through targeted resequencing. And so after this work, we've really moved everything completely over to the CRISPR-Cas9 platform, which I'll be going more into detail about. So this next phase of the project, we looked more carefully at CRISPR-Cas9 guides that would bind at CD40 ligand, and we identified additional ones that bound in the 5' UTR as well as within the beginning section of exon 1. And so we took the top 4 guides with the highest percentage of allelic disruption, shown here in the stars, and we analyzed them for off-target activity using guide-seq. And essentially what this is a cell line, in this case K562 erythroleukemia cells, are treated with our CRISPR guide along with a double-stranded oligonucleotide that contains a unique sequence that can be sequenced from using NGS. And so as you can see, we identified a guide with no off-target activity compared to the 3 other guides. And so this has been our optimal guide, cutting in 35 base pairs into exon 1. And so because we've always had a focus on eventually translating this into the clinic, we have delved further into off-target analysis of this particular guide. And so we did additional guide-seq analysis in K562s using both wild-type Cas9, which may have higher off-target activity, as well as a high-fidelity version that came out later. And you can see that there are no off-target sites in K562 cells. Based on a pre-IND meeting with the FDA, we were also advised to look for off-target activity in other cell lines. And so we proposed to use KG1A cells, which are promyeloblast cell line, and thinking that this is a human cell line with stem-like properties. And you can see that similarly to K562s, in multiple replicates using either wild type or high fidelity Cas9, that we identify no off-target sites. And that is in contrast to a VEGF guide that has known off-target activity shown below. We were also asked by the FDA to look at off-target activity in our cell product or our cell target. And this had been quite difficult because whenever you introduce double-stranded exogenous DNA sequences to primary cells, it's very toxic. And so when we attempted guide-seq in primary hematopoietic stem cells, there was so much toxicity that our data was not meaningful. However, in 2020, there was a publication in Nature Protocols describing an off-target analysis algorithm, which instead of using introduction of a unique oligonucleotide sequence, they leveraged the binding of specific DNA repair molecules to the site of double-stranded breaks. And so in this case, we're using chromatin immunoprecipitation to pull down for MRE11, which is recruited to the site of double-stranded breaks, which occur both on target and off target. And so we were able to apply this technology to primary human stem cells. And to our surprise, we actually identified this program identified many off-target sites. The on-target site is shown on the top in the red, and then the off-target sites are shown below. We were doubtful that these off-target sites were real, because as you can see, there's a significant degree of mismatch between these identified off-target sites and our on-target site. In addition, a lot of these mismatches are close to the PAM sequence, and in general, guide binding is intolerant to these type of changes. Nevertheless, we did look into these putative off-target sites through NGS using a ramp-seq protocol that was recently made available by IDT. And on the left, that tall blue bar is showing our on-target activity of around 70 to 80%. And you can see that when we compare mock treated, so the light blue, to the RMP treated samples, which are in dark blue, that the majority of these off-target sites are either below the limit of detection of the assay, or they are all similar to mock. And so this showed to us that the off-target sites identified by Discover-seq probably were not real. We've also looked at looked for general translocations by karyotyping peripheral blood stem cells that have been modified by RMP and identified no gross chromosomal abnormalities. And so moving forward with this particular guide and a AAV6 donor to deliver a codon optimized version of our corrective cDNA template, we moved this entire platform into T cells derived from XHI in patients. CD40 ligand is shown on the x-axis, and you can see that healthy controlled cells that are stimulated with PMA inomycin significantly upregulate their CD40 ligand expression. In the center are patient T cells that have not been treated and they do not express CD40 ligand. And on the far right are patient cells that have been gene modified by our CRISPR guide and Cas9 protein complex, as well as the AAV6 donor. We also wanted to evaluate for to make sure that the CD40 ligand expression that we had restored would behave physiologically. And so what we did was we kept gene modified T cells in culture and assessed them at 1 week intervals by re-stimulating them with PMA inomycin. And so if you focus on the light blue circles and the dark blue circles, you can see that cells that are gene modified are able to come to rest or go to rest and then be re-stimulated. go back to rest and be re-stimulated. So they behave in a physiologic manner. And finally, there's a clinical assay to look at the function of CD40 ligand by measuring its binding to chimeric mirroring CD40 in a flow assay. And you can see on the left that healthy controlled T cells bind well to CD40, as shown in that the pink peak on the right. And then patient gene modified T cells also bind properly to CD40. One thing that we noticed in many of our experiments in which we modified patient T cells is that the expression of restored CD40 ligand, as well as its binding to CD40, was always lower in intensity compared to normal T cells. And so this may not be a big deal in a situation in which you have high rates of gene editing and high rates of engraftment, but may become more important if we're not able to achieve the rates of gene editing that we're trying to achieve. And so we sort of, delved into seeing how we could improve upon our corrective donor. And the idea came up was, could there be some intron-mediated enhancement that we could add to our donor? And I won't pretend to understand intron-mediated enhancement, except to just very quickly say that, it's been known for a long time in many different organisms that the addition of an intron to a transgene can increase its expression. And so we started testing other donors using different codon optimizations, using different introns, using different lengths of introns, using different 3' UTRs, using different poly-A signals. And I wish I could say we did this very intelligently, but we sort of had to go through this brute force and sort of landed with our final donor that had the same codon optimization as our original donor, but included an intron. And so if you look at CD40 ligand expression on the top left, the healthy donor T cells are shown in the green. Our original donor is shown in the gray. We had a truncated intron that performed better than our original donor, and then a longer portion of the intron, which you can see behaves similarly to wild type. And you can see a very similar pattern for binding to soluble CD40 on the right. I also want to show on the bottom that the inclusion of an intron doesn't mean that the donor will express on its own, shown on the bottom. And then so the next section, I'm going to start talking about gene modification in hematopoietic stem cells. And so some things that we've had to keep in mind as we move this work into stem cells is that HSCs are long-lived and potentially lifelong. They're multipotent and transplantable, and this is why we're targeting them. However, they're also rare, they're delicate, they lose activity with ex vivo culture in that they differentiate, and they're subject to transformation from genotoxicity. And so when we're attempting to gene edit HSC, we need to be efficient, minimally cytotoxic, fast, and with high specificity. And so this is data in peripheral blood stem cells from healthy donors, and in cells that are treated with both the CRISPR and the AAV donor, you can see that in the light blue bar that about 25% of HSCs are gene modified without significant changes in viability or fold expansion short term. We also looked at the ability of gene modified stem cells to form multiple myeloid erythroid colonies using the colony forming unit assay. And so when gene modified stem cells are cultured on a methylcellulose media with certain cytokines for their differentiation, you can see that when we compare our gene modified samples, which are in the light purple, compared to mock treated and AAV donor only treated, that they result in the same distribution of different colonies. In addition, the colony forming unit percent of colony forming units grown per cells plated is relatively similar across the board, although a little bit lower for our gene modified cells. Because we're specifically interested in the ability of gene-modified stem cells to differentiate into T cells, since those are the cells that will be expressing CD40 ligand, we utilize the artificial thymic organoid system that was developed by one of our colleagues at UCLA. And essentially what this is a co-culture of stem cells, hematopoietic stem cells, with a murine stromal cell line that has been transduced to constitutively express a human notch delta-like ligand. And so that is essential for the differentiation of T cells. And so in this sort of 3D culture with a liquid air interface, we are able to achieve relatively good T cell differentiation that mimics that of a thymus. And so I'll walk you very quickly through the flow plots on the bottom left. If we gate out the NK cells and the stromal cells, and we focus on the NK negative and non-stromal cells, we can see that the gene-modified cells that are put through the ATO system can develop CD3 and are TCR alpha beta positive, so they develop a mature TCR receptor. If we focus on those cells, you can see that that we do have a population of double CD4, CD8 positives, mostly CD8 single positives, and very few CD4 single positives that can have a mature or naive phenotype based on RA and RO, shown on the bottom right. When we measure the percent gene insertion by droplet digital PCR, we can see that cells, peripheral blood stem cells that were gene modified, have on average 25% gene editing, and coming out of the ATO, so differentiating through the ATO, about 15% of T cells retain the gene editing 10 weeks in differentiation. And finally, we wanted to evaluate for the ability of gene-modified peripheral blood stem cells to engraft in NSG mice. And so stem cells are cultured for in pre-stimulation for 2 days, after which they are electroporated and transduced with the AAV vector, and then transplanted into NSG mice 24 hours later. And if to orient you, the gene-modified samples are in the blue bars, are in blue dots, and then the light gray samples are mock-treated cells. And as you can see, the percent engraftment in the bone marrow, spleen, and peripheral blood of mice are similar compared to mock. When we look at the lineage distribution of CD3, CD19, CD33, and CD56 cells, we see that they are relatively similar when we compare gene-modified cells with mock-treated. And then importantly, when we look for percent gene integration in the bone marrow, the mice that receive gene-modified cells retain about 8% gene marking in their bone marrow. And so I just very quickly wanted to sort of go over a timeline of where this project how far this project has come, and how much more it needs to go through before it reaches clinical trial. And so, you know, this work started very early on, comparing TALENs and CRISPR-Cas9, ended up with us deciding on CRISPR-Cas9 through multiple small funding sources, as well as a larger one through the California Institute for Regenerative Medicine. And we submitted an INTERACT package to the FDA in 2018, and met with the FDA in 2019. And based on their recommendations, submitted an application again to CIRM for a translational award, which we were given in late 2019. And so we're in the midst of our translational work, which involves scaling up all of the things that I have just shown you. So using GMP comparable critical reagents, as well as using many more peripheral blood stem cells that would be equivalent to what we might treat in an actual human. And we were hoping to meet with the FDA for a pre-IND meeting in 2022. This probably will be delayed until the end of 2022 or beginning of 2023 because of just because of the pandemic and delays in getting a lot of this work done. The hope is to have a IND meeting sometime in 2023, 2024, and to start an early phase clinical trial after that. And so now in our translational phase of the work, we have moved on to sort of validating a lot of our earlier work. And so now we're using GMP comparable CRISPR guide RNA, high fidelity Cas9 protein, GMP comparable AAV6 to deliver our donor cassette. And we've transitioned over to the MaxCyte electroporation system, which is scalable and can be used in a GMP setting. Cells that are gene modified are cryopreserved as we would expect them to be in an actual clinical trial before they're reinfused into patients. And so in some of our qualification runs, we have met our goals for our gene product. And so we're aiming for a viability of gene modified cells after thawing to be over 70%. Over 35% CD34 positive cells. Our goal is a gene editing rate of over 10%, and of course with no microorganisms in the final product. Because we are moving towards the clinic, we have also been able to recruit XHIM patients who have donated both T cells as well as peripheral blood stem cells for us to use in our work. And so this is one of our patients, and you can see we are able to edit his T cells like we have for our original patient, which we've used in our proof of concept work. And so his CD40 ligand expression went from essentially 0 to 29% in this experiment with restoration of CD40 binding. We were able to mobilize him with 5 days of GCSF, and in this case, without plerixafor. We were able to achieve a high yield of CD34 cells totaling 440 million. Below that, I'm showing the purity of the CD34 cells shown on the x-axis. And then we're able to take these collected CD34 cells, we're able to modify them in the lab in this patient, achieving a little over 20% gene editing, and with viability and fold expansion shown below. And so some observations that we've made from a lot of this work is that while we are able to achieve high rates of gene editing in vitro, there is a reduction in the efficiency of homology-directed gene correction in the more primitive HSCs. And this is in contrast to reported results with gene disruption, so such as at the CCR5 locus or BCL11A, in which when you're only trying to attain NHEJ or just knock out a gene, that percentage of NHEJ is pretty well maintained in vitro to in vivo, whereas that's not the case for HDR. And a limiting factor to efficient homologous directed repair in HSCs may be the DNA repair pathway activity of the cells. So as you may know, hematopoietic stem cells like to be quiescent. And so in a quiescent state, non-homologous end joining is favored in primitive HSCs. And so many times in order for us to have gene editing be efficient in HSCs, I mentioned earlier that we put them in 2 days of pre-stimulation, and that may cause them to differentiate. And so the limited efficiency of HDR and HSC may be a hurdle in moving this work toward the clinic. And so I'm just going to have a very quick diversion to a separate project that I worked on for a little bit, focusing on the BTK locus, which is responsible for X-linked A-gamma globulinemia. So this is just one slide, so it's a little side note, but it's not. I'm presenting this not so much to talk about gene editing rates for BTK, but to demonstrate that you could potentially design donors differently to harness different types of DNA repair mechanisms. So after creation of a double-stranded break, on the left, I'm showing that if you create your sort of classic donor that's flanked by homology arms, homology-directed repair can occur during the S and G2 phases of the cell cycle. And so, that works, but it's not ideal in the setting of a quiescent stem cell that we don't necessarily want to cycle. On the far right and in the middle, I'm showing two other types of targeted integration, termed homology-independent targeted integration, or PITI, and then precise integration into target chromosome, also termed pitch. And the idea here is that instead of targeting homology-directed repair, what if we attempted other forms of repair that can occur during other phases of the cell cycle? So on the far right is pitch, which is essentially integration using microhomology mediated repair. And then that tends to occur in the S phase of the cell cycle. And then in the center is homology-independent targeted integration, which is essentially end-trapping of a donor cassette into a double-stranded break. And so that end-trapping is sort of like an NHEJ event, which can occur in all phases of the cell cycle. And we postulated that this could be beneficial for targeting hematopoietic stem cells, which are quiescent. And so this was recently accepted into molecular therapy nucleic acids, and it's really just in vitro work for proof of concept. But on the left, I'm showing integration rates in the bulk population of peripheral blood stem cells, either using an HDR donor, a HITI donor, or a pitch donor. And you can see, I know there's a lot of data points there, but in general, the editing rates for HDR are significantly higher than the other repair mechanisms if you look at the bulk population of stem cells. However, we know that the cells that make up the peripheral blood stem cell population is very heterogeneous. And so if we gene modified a bulk population and then sorted for the immunophenotypic hematopoietic stem cells, which we defined as being CD34 positive, 38 negative, 45 RA negative, and 90 positive, you can see that the gene editing in those cells, which is shown in the red bar, is actually relatively equivalent across the three different repair mechanisms. And so, and theoretically, those are the cells that will be long-term engrafting cells. And so, we propose that, this could be a mechanism by which we achieve gene editing in primary stem cells, and it could be beneficial, particularly for diseases with gene cassettes that are a little too large for an AAV vector if you were to include homology arms. And so this essentially gets rid of the requirement for homology arms that take up space in an AAV vector. And then I'll finish up by transitioning back to CD40 ligand. And this is some very preliminary work that the story is not completed yet. But when we spoke with the FDA, we had proposed to include pediatric age patients in our initial clinical trials. And even though we're not ready for clinical trial, the reason that I proposed that early on is because the idea is that we need to develop gene-modified T cells from gene-modified hematopoietic stem cells. And the thinking was that could that process be more robust in younger individuals that have a larger or more active thymus compared to older individuals in which a lot of the thymus regresses. And so, they obviously do not like us to start off with children. And so, they reminded us that administration of investigational products in pediatric patients should not involve greater than minimal risk, or may involve greater than minimal risk if there is a prospect of direct benefit to the individual subjects. And so they suggested that we utilize the mirroring model of XHIM to demonstrate this. And so we've recently started using this mirroring model. And what we've done is, what we would like to do is essentially mimic everything we're doing with human cells, but purely with mirroring cells. And so in our hands, these CD40 ligand deficient mice seem to behave like the human model, which has been reported in previous studies. And so if we compare total IgG production in our CD40 ligand mice, we can see that it's significantly less than their healthy counterparts. When we immunize these XHI in mice, you can see that their healthy counterparts produce good amounts of TMP, KLH, IgG, whereas the CD40 ligand knockout mice do not. The difficult thing about moving this entire platform into murine cells is that even though the murine CD40 ligand locus also consists of 5 exons and 4 introns like the human, it's only similar by amino acid sequence to the human sequence by about 65%. And so what this means is that everything, we cannot use the same human reagents in the murine model. And so we've had to design murine-specific guide RNA for this part of the project. And so we sort of try to do this identically to how we performed design of guides in human cells. And so we started off by designing 3 guides that cut very closely to where the human counterpart cuts. We looked for off-target activity through guide-seq and identified 2 potential guides that we can use for additional studies. And then we've also started some initial work in murine lineage negative cells, which are essentially their hematopoietic stem cells, as well as in murine CD4 T cells. And so these cells are treated relatively similar with some differences in culturing time and conditions, but they are similar to their human counterparts. They are electroporated to introduce the CRISPR RMP. They are transduced with an AAV vector to deliver a copy of a normal murine CD40 ligand cDNA, and then there's further downstream analysis. And so on the bottom left, you can see that we can achieve relatively high rates of allelic disruption in both lineage negative cells, with achieving on average 50% gene disruption, and higher with murine CD4 T cells at about 80%. When cells are treated with both the CRISPR and the corrective donor, we achieve a little under 25% gene editing in lineage negative cells and around 20% for the murine CD4 cells. And so this is some of our very early work, and we're still sort of tweaking a lot of our protocols to see if we can improve the viability of the lineage negative cells that have been gene modified. And you can see on the right that that the viability of these cells is only a little over 50% after gene modification. And so historically, it's been very difficult to gene modify lin negative cells. And so we'll see if we can do better than this, but it's probably good enough to transplant back into the XHIM mouse model. And so hopefully there'll be new data to come, but not quite ready to present that yet. And so to conclude, the field of site-specific genome editing has shown tremendous progress in the last few years. This is ideal for the modification of genes that are tightly regulated. We've been able to achieve high rates of targeted gene modification at the CD40 ligand locus and in primary XHIM T cells and hematopoietic stem and progenitor cells. Gene-modified stem cells engraft and maintain gene marking at rates likely to ameliorate disease in vivo. And so a lot of our ongoing work still remains the translational portion of this project, which is scaling up and sort of getting things back running after the pandemic. And then, of course, our hope is to have the murine HSPC editing be completed in parallel to the translational work, so that when we are ready to meet with the FDA for a pre-IND meeting, we can present them with both parts of this data. So I want to thank you very much for having me here today. Thank you so much for taking the time to be with me. Of course, I have to thank my mentor and my group that works with me on a daily basis. And of course, I'm grateful for my funding sources. So thank you very much, and I'm happy to take any questions.
Thanks, Carolyn. Guys can either put your questions into the chat or just ask them directly. Great talk. Thank you.
Thank you.
Hey, Carolyn. You had put that your target for the trial that you submitted to the FDA was 10%. I was curious kind of how you guys came up with that, and You guys achieved 19%, but that's for the actual editing rate.
Mhm.
You'd anticipate kind of a reduction in engraftment.
Right.
So what do you guys think in terms of what do you eventually need in terms of the cells that engraft and actually make CD40 ligand?
Yeah. So that goal that we set was, a relatively low goal. I think that, you know, if we look at female carriers of CD40 ligand mutations in which they have skewed lionization towards the defective gene, they tend to be asymptomatic down to 10% normal T cells. And so, our goal, we've always said our goal is to achieve 10% editing in engrafted stem cells. Our goal in vitro, of course, should be to achieve higher rates than that, knowing that the rate will drop in vivo.
Thank you.
Hi, Carolyn. Great talk. I have a kind of a conceptual question. I don't know, I don't know if you'll be able to answer it, but I don't mean to be a jerk. But I guess my question is kind of just about CD40 ligand on T cells. So when you overexpress it, you get this lymphoproliferation phenotype. Does that require B cells, or is it something about the ligand itself that's signaling in the T cell, in a cell intrinsic way?
Mm. I could be wrong. I've always understood it as it requires B cells.
Yeah, I think that's what we would generally think, but
Yeah. But I guess I don't know. I haven't studied that and I don't know for certain.
okay. Yeah, sorry about the question. It's like
No, not at all.
And
Good to think about.
neither of those groups that published those early studies looked at whether B cells were required. So we don't really know the answer to that.
Yeah.
Hi, Carol.
Jump up there.
Just following up on the CD40 ligand, you mentioned that having intron-mediated enhancement of CD40 ligand expression. I'm just curious what intron you added into the CD40 ligand and how you made that fit. I was looking at some of the introns in CD40 ligand, and they're 1800 base pairs and stuff.
Yeah, good question. So we ultimately landed on intron 4, and we did not include the entire intron because it doesn't fit. And so we sort of took sections of the intron that in genome browsers appear to be highly conserved among species and appear to have DNA methylation sites, or I don't remember all the specific criteria that we did, but it was a little bit hand wavy, right? It's hard to define exactly what section was important, and so that's why I showed a little bit of data. We had a truncated one, and we ultimately used something a little bit longer. So it does require a little bit of testing to determine that.
Thank you.
You're welcome.
Caroline, I have two questions. One, I assume that the product of CD40 ligand you generate forms trimers, a normal trimer formation that then locates into the cell membrane if the cell is activated.
So is your question, do I know that they form trimers?
Does it naturally form like the natural CD40 ligand that does the product which you generate forms trimers normally?
I guess, how would I assess for that?
I think the fact that it binds to CD40 would indicate to me that it is a trimer.
Okay.
But then what happens to the mutated gene that is still left over? Is that silenced?
That's a great question, because we have always assumed that it's silent. It remains silenced. But
yeah, that is the important question, because most mutated CD40 ligands have actually a product,
Uh-huh.
either truncated, or an amino acid substitution, and still will form trimers with the gene corrected. And so unless the natural mutated CD40 ligand is silenced, you would have a mixture in your trimer formation of the gene corrected and the non-corrected product that would be generated in the same T cell. And then you would have a mixture, between normal and abnormal CD40 ligand proteins that form a trimer.
So I guess the way to test for that would be to look for downstream things, right? To look at the function of the CD40 ligand, like class switching or something else further downstream.
And that's why it's so important to do this first in mice. But I would assume that the fact that you see very good binding of the CD40 ligand, the corrected, with the CD40 Ig protein, that would tell me that probably there is not a diminution due to the utilization of the abnormal CD40 ligand.
Yeah, so I mean that's a good point to think about, and we had attempted to look at class switching in vitro, and we were never successful. Really, we just couldn't get the assay to work, and but hopefully some of our mirroring work can answer some of those questions. Yeah.
I think one part of the answer to that, Hans, is that when Carolyn edits upstream and introduces a cDNA under the control of the endogenous promoter, that the endogenous transcript is also not normally transcribed there because you edited the first exon, right?
That would be the answer. If that is,
I don't think that's
essentially eliminated.
Yeah, I think it eliminates most of that risk. Are there any questions from trainees or other folks? It'd be great to.
I have a couple of questions regarding the mirroring editing. First off, great talk.
Thank you.
Very interesting. But regarding the mirroring editing, judging from your cartoon, it looks like you use a Lonza nucleofection system. Is that correct? Am I correct in saying that?
No, we use the neon.
You use the neon, okay. And then a follow-up question: have you noticed any viability issues with the Lonza? Is that why you use the neon, or?
No. So why did we start with the neon? I think, we started off with the neon because I had used the neon for some of my other work, not for CD40 ligand, and found that it seemed to be more gentle on cells. So this is totally just sort of, it may not even be true. That was my observation working with other sorts of cells that are difficult to electroporate. And so we just sort of started off with that, and we seem to achieve the highest. And now that we've compared it to the Lonza, we seem to achieve the highest rates of gene editing still with the neon. We've also compared it to other electroporation platforms like the CIIT, and even though we can achieve similar rates of editing, they still trend lower. And so we've sort of stuck with the neon for now.
Sure, that's thank you for that. We've noticed kind of a similar phenomenon lately, so wanted to double check. Thanks.
It's good to have confirmation.
Just a random question. CD40 ligand comes up on activated platelets. Have you guys looked? Does your construct actually activate the right way?
We have not looked. We certainly can. Yeah.
Not sure what its function is in that role, but it definitely stops some of the other therapeutic, monoclonals.
Yeah, we can do that.
Hey, Carolyn, I have a quick question. Thank you for your presentation, first of all. I'm wondering if you guys have incorporated any kind of additions to your editing platform to improve engraftment or editing within the long-term HSCs, things similar to like the Naldini or Malik groups?
Yes. Yeah, great question. So we did, we did look at some of, so for example, the Malik group uses a different media, they have different additives, they use different amounts of cytokines. And we did do a very small pilot experiment comparing sort of our base media, so just ex vivo with stemflit TPO, to the Malik group's SFEM2, and also added UM171 and SR1. As well as the I53, the version that they used. And, to be honest, in this very small pilot experiment, to be honest, the in vivo outcomes were similar across the board. Maybe we needed more mice. I don't know. So we sort of, we didn't pursue it much more, although there are other groups sort of in the larger lab that I'm a part of that are looking at different media comparisons and looking at different additives that can be, used to improve engrafted, gene mark cells.
Thank you.
I have another small. Oh, sorry, Bruce. Do you want to go?
No, go ahead. I'll go after you.
I have another kind of small question. You guys have explored HIDI, and standard HDR at the BDK locus. Have you looked across other loci? I mean, I understand the point about there being differences in quiescent cells versus activated cells and things like that, but is there loci specificity to the effectiveness of the various techniques, or do we know anything about that?
There very well might be. There very well might be. So, for example, there in the HIDI work that I presented, that is the only guide that we have in which it only works. The cutting only is highly efficient when paired with Cas9 mRNA. In general, cutting is more efficient with Cas9 protein. We haven't been able to figure it out. We've purchased new stocks of guide. We've did everything we can to sort of make sure we're using the right reagent, but it's the only locus that does that.
Well, BDK is definitely weird, for knockout, for sure.
Yeah. So sort of stemming from that, I wouldn't be surprised if there are significant, differences between different gene loci.
Thanks. Again, great talk.
Thank you.
Excellent talk. It was quite nice to hear that. Going back to this question of quiescent stem cells,
Mm-hmm.
and the level of cutting, have you played around with the conditions at all with the quiescent cells to see if you can increase, other than some of the systems that you use, the HIDI and the others?
You mean in addition to like maybe additives that keep them quiescent?
Yes, absolutely.
Not beyond, so, we played around with UM171 and SR1.
Right.
We did see differences in the immunophenotypic sort of profile of our stem cells, but did not see differences in editing overall.
So it was still the same proportion of editing.
Yeah.
Was it when you did that, was it restricted to the more quiescent cell population? Do you know?
What was restricted to the quiescent cell population?
When you added the additives, I was curious, looking back.
Right. So it increased. Okay. So maybe I should take this back. So it increased the proportion of the immunophenotypic HSCs. And then the percentage of gene editing was maintained. So I guess you can argue that if you increase the percentage of those cells, then the absolute number of gene modifiers
would increase.
Yeah, it would increase. It was mild though, and we never sort of scaled it up further, and it may be worth looking into.
And given the genotoxic activity that's seen as you activate, the longer you keep them activated, when you did some of your work, did you see more genotoxic when you had longer periods of time in culture? Was the genotoxicity increased at all? And did you see any chance of additional cutting or off-target events?
Yeah, so we have tried longer pre-stimulation. So for example, the Naldini group, I think now many times, pre-stimulates for 3 days. They
Yes.
have other additives involved, and so we did not include the additives that they use, but we actually saw lower gene editing when we cultured for 3 days. Surprisingly.
Interesting. Okay, interesting.
So, and then we've also tried culturing for shorter periods of time with HITI, thinking that the cells don't need to cycle if you're just end capturing into the double-stranded
Right.
break. It didn't seem to pan out. It didn't come to fruition.
Interesting. That was really nice work. Thank you.
I'll take the prerogative for one last question if there aren't any others. But we're our group and yours are using AAV as the HDR donor predominantly, right? How much of the engraftment issue do you think is AAV mediated as opposed to all the other issues that you discussed?
I feel like it's all AAV mediated. So we have, we've made some attempts at using non-viral DNA donors, and I think that's still an incomplete story because we really just started looking into those and have found that oftentimes we don't get significant rates of gene modification. But it may relate to sort of what Marson's group has reported, and that maybe it just doesn't get into the cell well, and you need to include Cas binding sites to sort of help shuttle the donor into the nucleus. And so it's something that we're looking into, but we don't have any results from that, and really have had very low rates of modification with non-viral donors.
And we would argue that you see a lot of toxicity with the non-viral donors. That
yes, I would agree with that. Yes.
The AAV impacts are hard to understand, I think.
Yeah.
But I agree with you. I think it's a major aspect of this that's like, we need it, but we also need to understand what's happening.
Right. And one thing that we've also observed is that there are significant differences in how AAV performs or how toxic it is depending on who produces it. And so I think that's also a worry for me as we sort of chug along towards the clinic, is that a lot of the AAV production by companies is not standardized. And a lot of times they don't share with us exactly what they do, and that may be an issue in the future.
And your GMP like AAV is, you're not really able to control that process?
Exactly. No. Yeah.
Rich, do you have another question?
I was just going to comment that, is it presented like by MHC? In such a short turnaround, could be immunogenic, right? I don't know. Is there any data on that? Because it's testable.
I don't know.
I don't think there's any data on that, but it could be cell intrinsic or extrinsic or both.
Right.
Not sure if the time's up for questions. I have another question.
We actually have 30 minutes after the seminar, not 30 more minutes, but until 1:30. So yeah, this is why we have the 30-minute period so you guys can ask questions. So go ahead.
Yeah, just a question. So you tried pitch, you tried HITI, you tried homology directed recombination. Have you ever tried a homology mediated end joining where you cut off the ITRs outside of the homology arms with guide sites, and then looked at NHEJ essentially of your gene?
That's how we did the HITI experiments in peripheral blood stem cells.
So you maintained the homology arms?
Yes.
With the, okay. So, thank you.
May I ask a practical question? So CD40 ligand is not required for making CD4 positive cells. And you see that in carrier females, they have 50-50 average. So to get this in, you are using a new approach. You know, before in X-linked SCID, for instance, there is no problem. The corrected hematopoietic stem cells will immediately go into T cells. The same thing is true with Viscard. But here you need to get many corrected stem cells. And so are you prepared to condition the patients with a myeloablative protocol?
Yeah, that's a great question, and one that I struggle with, and actually, and Alex and I have talked a little bit about that earlier today, and is that, you know, I think that they do need myeloablative conditioning. If we look at the, you know, for example, if you know, if we focus on the fact that we need T cell reconstitution of from gene modified stem cells, if we compare it to the lentiviral trials for XCGD and for WAS, from what I could be wrong, but from what I've looked at, the gene marking in the T cells is oftentimes tenfold lower than in any of the other cell types that are assessed. Yeah. And so my worry has always been that we can gene modify the stem cells, but we won't get significant T cell differentiation. And so you know, when I think about the conditioning regimen, I do think that they need something that is myeloablative, and we also need to create room in the T cell niche.
Yeah, and the experience from hematopoietic stem cell transplantation also support that, because these patients do not as well with hematopoietic stem cell transplantation as Viscards, for instance, or SCID patients, ADA deficient patients. So I think that makes me also think that this is a very unique model. But it's important to see if we can get better than hematopoietic stem cell transplantation in hyper IgM syndrome.
Great. Well, any last questions? Carolyn, thanks for answering all the questions.
You're welcome.
Actively, and a great talk. I think everybody learned a lot from your talk. If not, we'll say thank you. And then I think you and I have I think in 10 minutes we can call into the WebEx. And
okay, sounds good.
Yeah.
I think.
Thanks, Caroline.
Thank you.
Thanks, everyone.
Thank you, everyone. Bye-bye.
Bye.